Okay, so hi everyone. I'm very glad to be here today to present to you a work I did during my PhD. So now I'm a postdoc at the Institut Pasteur in Paris, but before that, I was in Rennes and Brussels. And I worked on genome assembly and metagenome assembly. So for those of you who are not aware exactly of what that is, genome assembly, so the problem is you have genome, you sequence them, you obtain reads, and the problem of genome assembly is stitching all those reads together to obtain the genomes. And genome assembly and metagenome assembly is really a computationally intensive task. And something I really liked during my PhD was launching the computation on a Friday night, going away on the weekend, and coming back on the morning, and it would be done. And it would feel really good because I would have watched Netflix all weekend, and I would come back on Monday and think, that was a productive weekend. And this worked very well until I tried to assemble this gut metagenome, which is a hi-fi data set of 250 gigabytes. And this was very hard for me because I came back on the Monday morning and it was not done. Because the assembler I used at that time, Metafly, took 4 days and 250 gigabytes of RAM to assemble this data set. Hi-Fi ISMB took 11 days and almost 500 gigas of RAM. And so this made me feel bad because I did not have my little thrill on the Monday morning of feeling like a productive weekend. But it's not only a problem of me, it's also a problem that metagenomic datasets in particular are still going to grow in the future years. And here we already are at 500 gigas of RAM for 11 days. It's already a big computation that not everyone can do. So we really need to scale up to have faster assembly. Thankfully, just around that time came out MetaMDBG. So I don't know if you were there in the microbiome track yesterday, but Christopher Quince presented it extensively, super interesting, and extremely fast. 19 hours, only 10 gigas of RAM for this data set. Cool, my weekend was saved. I could go back to watching Netflix and feeling productive. And the trick of MetaMDBG is that instead of running computation on the whole reads each time, it only samples some minimizers, some k-mers, and it runs the expensive computations, which is stitching all the reads together, only on this reduced minimizer dataset. And this makes it much more efficient than other assemblers. And this is a very well-known trick that has been implemented in many other tools, like for alignment, for phylogenetic compression, and everything. The problem was that when I analyzed my data, I actually saw that different strains that were present in my sample got collapsed, which means that, for example, here we have those 2 strains differentiated by 1 SNP. And after assembly, I only recovered 1 strain, which can be very limiting depending on the data set, but strains are known for being having potentially very different phenotypes. For me, it was very problematic. So I was stuck there with 2 problems, one being assemblers that are too slow and too memory intensive, and the other one being MetaMDBG, which is very fast, but collapses the strains together. And the work I'm going to show you today is about how we can actually solve both of these problems at the same time, having both a fast and a strain-aware assembler. So my first thought was, maybe I can just tweak a little bit MetaMDBG, spend a few days working around the code and solve this problem. But no, it doesn't work that way because the problem is really fundamental. It's if you have 2 reads differentiated with 1 SNP difference here, well, 2 MetaMDBG, if this SNP falls between the minimizers, well, they look exactly, the two reads look exactly the same to MetaMDBG, so they will be assembled together. And there is really no way around that if we sketch if we sketch the reads using minimizers. So then I ask myself the question, is k-mer subsampling really the only way to sketch sequences? I would say that it's really the one that's mostly used today in genomics, but is it really the only way? And I read this article at that point, which was a lucky coincidence, that talked about mapping-friendly sequence reduction. So it was somehow related to homopolymer compression, and it was not presented as a sketching technique, but actually it can be used as a sketching technique, and I will show you how just now. So let me present to you what mapping-friendly sequence reductions are. To compute a mapping-friendly sequence reduction, you need a function that maps a k-mer, here I have a 10-mer, to either a base or a special empty character. So here is an example of such a function, but basically all functions work with just a small constraint for a reverse complementarity. And so you have a sequence to sketch. And to sketch this sequence, you look at all the k-mers, and so you begin by the first k-mer, you run it through the function, and it outputs an A. So you append an A to the sketch. Then you move on to the second k-mer, you run it through the function, and it gives you a special empty character. So you don't append anything to your sequence. And so on and so forth. You slide the k-mer across the whole sequence until the end. And here you have the sketch. The sketch is just the bottom sequence. OK. So that's somehow an extension of homopolymer compression. OK, so that's an intriguing way to transform a sequence. But let's see what we can do with it. Well, first of all, what we can observe that there are two kind of important hyperparameters in this compression. It's the compression ratio here, 0.2. So in 80% of my case, the function gave an empty character, and that means that my sketch will be 5 times smaller than the input sequence. And the other hyperparameter is the order. So that's a bit more subtle, and I'll discuss this later. So the order is the size of the k-mer window we slide around the sequence. So MSRs are called like that, and the important part is really the mapping friendly. And that's really the key property that make MSR really interesting. Is that basically you have an equivalence between the fact that the original sequences align and the fact that the reduced sequence align. It's not a perfect equivalence because we lost some information, but it's mostly an equivalence. Well, you can see here, of course, if we have 2 times the same sequence, they will be sketched to the same sketch, so the sketch will align too. And this is a very useful property for many things. It means that, and especially for assembly. So if you look at how we can run assembly using MSR sketches, so you take all your reads, you reduce them using MSR sketches, and then you can assemble the sketch. And that's because if read 3 and read 1 overlapped, aligned in their original space, well, then their sketch will align too. And that means that instead of assembling the original reads, you can just assemble the sketch. And once you have assembled the sketches and you know that read 3 overlaps read 2 read 3 overlaps read 1 overlaps read 2, you can go back to the original reads and deduce that well, you have first read 3, then read 1, then read 2. And this whole process is actually much faster than regular assembly because the computationally intensive part, the difficult part, is really assembling the sketches, it's stitching all the reads together. And here we're doing it with much shorter reads. So it automatically goes much faster. But you remember my second problem. So fast assembly is something. My second problem was that I lost variants and I lost SNPs. And this sketching method is actually quite good for that. So if we look at these two sequences, which are only different by one SNP, and we run the MSR sketching on them, well, up until the SNP, they will be identical, of course. But then around the SNP, you will have different k-mers up and down on the two sequences, and so they will output potentially different sequences. And you can run that until the end of the sequence. And once again, at the end of the sequence, you will have the same k-mers sliding around the same sequence. And so we will have identical sketch again. And actually, MSRs, if you look at the keep unamplified SNPs, if you look at the SNPs before, it was one base on a long sequence. And the SNP after is actually made of two bases in a much shorter sequences. So the SNPs have been amplified. And that works when we have a k-mer length here of 10 that is sufficiently long, which means that you will have enough k-mers around your SNP so that at some point you will output some bases. And you have no reason to output the same bases on the top and bottom sequences, so your two sequences will be different statistically. But that's super useful because it means that we can sketch reads, and that reads that are a little bit different won't be sketched to the same sketch, which was the problem of MetaMDBG. And so I've encapsulated all this assembler in what's called the Alice assembler. It's named like that after Alice in Wonderland, because she, at some point, she tries to go through a small door, and this is an analogy for a big data set on a small computer. And she takes a drink me potion to shrink. This would be the sketching method. And so then she can do the difficult part, going through the door, assembling the reads in reduced mode. And then she inflates on the other side of the door. And so there is a first version that's available on GitHub. And well, you're welcome to try it out. It's becoming kind of stable now. So I think it's not too buggy anymore. And a nice thing also is that you can really use any kind of assembly here. I didn't have to recode any assembler. I just coded something that reduced the reads. I ran it through an existing assembler, and then I inflated the reads. But it was very little work in terms of assembly. Now I'm going to show you some results. I'm not going to show you a lot of results because evaluating assembly is a little bit tricky and you need a lot of metrics and it's a bit tedious. So I'm just going to show you some highlights, and you will see in the upcoming preprints more comprehensive evaluation. But the first dataset I evaluated on was the Zymo Biomic Gut Microbiome Standard, which is a mock dataset that contains 5 strains of E. coli. And if you look at the percentage of these strains that are recovered by MetaMDBG, you see that some strains there is less than half of the genome that is actually outputted in the final assembly. And in the Alice assembler, it's much nearer 100%. So first point is really good. The strains are not collapsed. And that was what I was aiming for. Second thing that I was aiming for is having something that goes fast. And so here I'm going to show you the example with the human gut metagenome dataset, 250 gigabytes. And so Alice assembled it in only 5 hours and 10 gigas of RAM. So I was very happy about that. And now I can not only go on weekend and watch Netflix all weekend, I can even do it overnight and come back on a Thursday morning and say, this was a productive night. So I'm very, this made my PhD much easier after this. MSR is not magic. There is a dark side to MSR. I've told you how it amplified SNPs. Well, if this wasn't a SNP, but a sequencing errors, it's amplified too. So this won't be adapted for erroneous reads. This is really adapted for low error profile reads. And I'll finish by saying that in the future, especially metagenomic datasets will continue to grow, and that we really need efficient algorithms to assemble them. So there is an assembler that's available online already. MSR sketches are a type of sketch that are that have very intriguing properties and quite different from K-mer subsampling, because they keep and amplify the differences between the original sequences. And their second very interesting property is that they are themselves sequences, and since they align like regular sequences, they can be used as such in many applications. And as a perspective, I would say that there can probably be many other uses for MSR sequence sketching besides assembly. And there are a lot of things I didn't explore in MSR sketching. For example, this function, the choice of this function is probably super important, but I don't have. I explored only one of these functions basically. So I will finish my talk here, and please ask your question if you're interested.
So I see that we have some questions. So microphone number 1 is ready.
Hi, that's very promising. That's exciting. Yeah, that could be a game changer. I think that's awesome. So I'm just wondering, first of all, did you test this on short reads data, and how does that come out? Specifically, short reads metagenomes, and I'm also wondering about how. I mean, what are the odds of not picking up, the SNPs, because it's going to allow this to you? And I'm assuming that those ranges on the right have been optimized benchmark. And can you comment on the
Yes.
probability of missing, a SNP?
Okay, so the first question, short reads. Well, the thing is, since the point is reducing the size of the read, if you reduce a short read by a factor of 20, you will obtain 7 bases, and you will never be able to assemble or do anything with 7 bases, right? So you're a bit limited with that, but it will, elsewise it will work well. It's just that you're not going to have a good compression factor. For your second question, yes, I have computation. It's going to be in the preprint, and basically, but it really depends. Actually, it really depends on the order here, the 10. If you take a longer 10, you're less likely to miss SNPs, and if you take a shorter 10, you're more likely to miss SNPs. But then the longer you take, the more you amplify the errors and the SNPs. So that's kind of a trade-off.
Okay, thank you.
But it's exponential in the order. So.
Okay, so now microphone number 2.
Thank you. Very interesting talk. So first, simple question. It's right slide exactly. So am I correctly understand that you can take any number of beans, not just 4 beans as here, and have you experimented with different number of beans, and or you just fixed that 4 you can name is the same and it's familiar. What changes if the number of beans is changed?
So you mean the number of the size of this alphabet, right?
Yeah.
Yeah. So it's a good question. The reason I chose 4 is because then you can just run it through any existing software and it works.
I see.
And no, I haven't explored, but I think that's really definitely an interesting thing. It's very subtle, I think, the way it's changed, but I don't know yet. Yeah.
Okay, and the second question was, of course you said that if you have errors in read, then errors will propagate. But suppose you have just not an error, but just a variant, and it happens to be close to the end of the read, and due to the propagation, you will get a different assembly. How often this happens? That because your error propagate somewhere to the end of the strand, you get a completely different assembly. But
yes, definitely. So that really depends on what you really have to think of what you want. So in my case, I wanted things to be completely different because I just wanted to assemble completely separately the reads. But indeed, if you do, for example, SNP calling, you may be very careful with the function. I think changing this function is really the key thing, so that one SNP either becomes a huge difference or stays a small difference.
Okay, thank you.
One more question from microphone 2. Yeah.
Great talk. I have a question about how do you choose the hash function, because I guess MSR is kind of optimized for, I mean, mapping exactly, but there are different kinds of modes in MSR. So did you try different hash functions and get the best one, or default one?
So what I was trying to do there was to have a hash function that was very chaotic, and so that if I had a snip, I want to have no chance of having the same hash or a close hash. I want to have a very chaotic hash. But on the other side, having a non-chaotic hash which maps similar k-mers to similar hashes could also be very interesting to deal with errors. And it really depends on what you want. And here, it's since I wanted to separate very starkly things that were even one snip apart, I took this the most chaotic hash function I found, which was I used anti-hash, I think, at the end, which is supposed to be chaotic.
Thank you.
I have one quick online question. What about large genomes?
Yeah, large genome can be definitely compressed sketched using that, and I think that could be definitely one application because you don't expect many errors to be in assembled genomes. So compressing large genomes using MSR sketching, I think, could be a really interesting idea.
Okay, so let's thank the speaker again.